Hi there, how are you? Welcome to another biology class. Today we will continue what we have started last time, part 2 of the discussion on chloroplast. Before we start, make sure you have subscribed to this channel and please be kind to like and share this video. Alright then, let us begin. Glaucophyta. Usually the endosymbiosis event is considered to have occurred in the Archaeoplastida, within which the Glaucophyta being the possible earliest diverging lineage. The Glaucophyte chloroplast group is the smallest of the 3 primary chloroplast lineages, being found in only 13 species, and is thought to be the one that branched off the earliest. Glaucophytes have chloroplasts that retain a peptidoglycan wall between their double membranes, like their cyanobacterial parent. For this reason, Glaucophyte chloroplasts are also known as moroplasts, besides cyanoplasts or cyanals. Glaucophyte chloroplasts also contain concentric unstacked thylakoids, which surround a carboxysym and icosahedral structure that Glaucophyte chloroplasts and cyanobacteria keep their carbon fixation enzyme ribisco in. The starch that they synthesize collects outside the chloroplast. Like cyanobacteria, Glaucophyte and rhodophyte chloroplast thylakoids are studded with light-collecting structures called phycobilisyms. For these reasons, Glaucophyte chloroplasts are considered a primitive intermediate between cyanobacteria and the more evolved chloroplasts in red algae and plants.
Rhodophysa, red algae. The rhodophyte, or red algae chloroplast group is another large and diverse chloroplast lineage. Rhodophyte chloroplasts are also called rhodoplasts, literally red chloroplasts. Rhodoplasts have a double membrane with an intermembrane space and phycobilin pigments organized into phycobilisyms on the thylakoid membranes, preventing their thylakoids from stacking. Some contain pyrenoids. Rhodoplasts have chlorophyll and phycobilins for photosynthetic pigments. The phycobilin phycoerythrin is responsible for giving many red algae their distinctive red color. However, since they also contain the blue-green chlorophyll and other pigments, many are reddish to purple from the combination. The red phycoerythrin pigment is an adaptation to help red algae catch more sunlight in deep water. As such, some red algae that live in shallow water have less phycoerythrin in their rhodoplasts, and can appear more greenish. Rhodoplasts synthesize a form of starch called fluoridine starch, which collects into granules outside the rhodoplast, in the cytoplasm of the red alga. Chloroplastida, green algae and plants. Green algal chloroplasts are characterized by their pigments chlorophyll A and chlorophyll B, which give them their green color. The chloroplastida chloroplasts, or green chloroplasts, are another large, highly diverse primary chloroplast lineage. Their host organisms are commonly known as green algae and land plants. They differ from glaucophyte and red algal chloroplasts in that they have lost their phycobilisyms and contain chlorophyll B instead. Most green chloroplasts are, obviously, green, though some aren't, like some forms of Haematococcus pluvialis, due to excessive repigments that override the chlorophyll's green colors. Chloroplastida chloroplasts have lost the peptidoglycan wall between their double membrane, leaving an intermembrane space. Some plants seem to have kept the genes for the synthesis of the peptidoglycan layer, though they've been repurposed for use in chloroplast division instead. Most of the chloroplasts depicted in this article are green chloroplasts. Green algae and plants keep their starch inside their chloroplasts, and in plants and some algae, the chloroplast thylakoids are arranged in granostacks. Some green algal chloroplasts contain a structure called a pyrenoid, which is functionally similar to the glaucophyte carboxysym in that it is where rubisco and CO2 are concentrated in the chloroplast.
Helicosporidium is a genus of non-photosynthetic parasitic green algae that is thought to contain a vestigial chloroplast. Genes from a chloroplast and nuclear genes indicating the presence of a chloroplast have been found in Helicosporidium even if nobody's seen the chloroplast itself. Polynella chromatophyra. While most chloroplasts originate from that first set of endosymbiotic events, Polynella chromatophyra is an exception that acquired a photosynthetic cyanobacterial endosymbiont more recently. It is not clear whether that symbiont is closely related to the ancestral chloroplast of other eukaryotes. Being in the early stages of endosymbiosis, Polynella chromatophyra can offer some insights into how chloroplasts evolved. Polynella cells contain 1 or 2 sausage-shaped blue-green photosynthesizing structures called chromatophores, descended from the cyanobacterium Synechococcus. Chromatophores cannot survive outside their host. Chromatophore DNA is about 1 million base pairs long, containing around 850 protein-encoding genes, far less than the 3 million base pair Senecacoccus genome, but much larger than the approximately 150,000 base pair genome of the more assimilated chloroplast. Chromatophores have transferred much less of their DNA to the nucleus of their host. About 0.3-0.8% of the nuclear DNA in Polynella is from the chromatophore, compared with 11-14% from the chloroplast in plants.
Many other organisms obtain chloroplasts from the primary chloroplast lineages through secondary endosymbiosis engulfing a red or green alga that contained a chloroplast. These chloroplasts are known as secondary plastids. While primary chloroplasts have a double membrane from their cyanobacterial ancestor, secondary chloroplasts have additional membranes outside of the original two, as a result of the secondary endosymbiotic event, when a non-photosynthetic eukaryote engulfed a chloroplast-containing alga but failed to digest it, much like the cyanobacterium at the beginning of this story. The engulfed alga was broken down, leaving only its chloroplast, and sometimes its cell membrane and nucleus, forming a chloroplast with 3 or 4 membranes, the 2 cyanobacterial membranes, sometimes the eaten alga's cell membrane, and the phagosomal vacuole from the host cell membrane.
The genes in the phagocytosed eukaryote's nucleus are often transferred to the secondary host's nucleus. Cryptomonads and chloracneophytes retain the phagocytosed eukaryote's nucleus, an object called a nucleomorph, located between the second and third membranes of the chloroplast. All secondary chloroplasts come from green and red algae. No secondary chloroplasts from glaucophytes have been observed, probably because glaucophytes are relatively rare in nature, making them less likely to have been taken up by another eukaryote.
That is all for today, folks. One last thing. Before you go, make sure that you have subscribed to our YouTube channel and be kind to like and share this video. This video is edited by Brooke Solomon, MSc in Genetics. I will see you in the next video. Goodbye.